Good morning, children. I explained you the three types of solutions yesterday. Isotonic solution, that is when the solution outside the cell and inside the cell, their concentration is same. So there will be no movement of water molecules from outside to inside or inside to outside. No osmosis. The second type of solution is hypotonic solution. When a solution outside the cell is having lower concentration of solute, that is, it is a dilute solution as compared to the solution inside the cell. That is, inside the cell, the solution is concentrated. In this case, the water molecules from outside to inside of cell will take place, hence end osmosis occurs. Third type of solution is hypertonic solution. A solution having higher concentration of solute molecules outside the cell as compared to the solution inside the cell. In that case, the water molecules from within the cell will move out and thus exosmosis will take place. To understand the above three conditions and process of osmosis, we will take human red blood cells. We'll place them in three different dishes. In one dish containing isotonic solution, second dish containing hypotonic solution, and the third dish is containing hypertonic solution. In dish A, in this setup A, when we'll put the red blood cells, human red blood cells, we'll find the shape and size of the red blood cell remain unchanged as there is no net osmosis. In setup B, the contents of red blood cell is hypertonic as compared to the outside solution. So water molecules from outside, that is hypotonic solution, will move inside the red blood cells which is hypertonic in nature. In this case, the red blood cells swell up or may even burst due to end osmosis. In the third setup C, we will see the red blood cells shrink. This is due to exosmosis because the outside solution is hypertonic, more concentrated than the solution inside the red blood cell, which is hypotonic. So the water from inside the red blood cell will move out and this will result in shrinkage of cells. Similarly, we can put plant cell also in these three different solutions. You can see the figure. This is a plant cell which is placed in a beaker containing isotonic solution. No change in the shape and size and structure of the cell because the solution inside and outside the cell their concentration is same. Then the plant cell is placed in the second setup, that is containing hypertonic solution. In this case, the outside solution is hypertonic, more concentrated than the solution inside the cell. So exosmosis will take place. Water from inside the cell will move out. See the arrow? Thus it results in the shrinkage of the cell. The protoplasmic cell membrane, that is the protoplasmic content leaving the cell wall, will shrink. In the third setup, we'll place the plant cell in a beaker containing hypotonic solution, that is more of water molecules outside, and inside the cell sap is having less of water molecules. It is dilute. the cell inside has less of water molecules, so it is concentrated. So water from outside will move inside due to end osmosis. This will result in the swelling of cell. The cell becomes, when the cell swells up, we say it is turgid. Cell becomes turgid. So this is how we can explain the shape, size and structure of the, taking the example of plant cell and a human red blood cell. Now we'll do the significance of osmosis. We all know the cell sap of root hair is hypertonic as compared to soil water. That is why the roots of plants absorb water from the soil due to end osmosis. Then the water absorbed by the roots move in the upward direction from cell to cell due to osmosis. Why it happens? Because of the fact that large amount of water is lost by the leaves of plant due to transpiration. Those cells which lose water as water vapor due to transpiration, the cells become hypertonic as compared to the lower adjoining cells. So water from the lower cells will tend to move towards the upper cells from where the water has evaporated. So osmosis takes place from hypotonic side to hypertonic side. This accordingly water keeps on moving in upward direction. Then the stoma in the plant cells open or closes in response to the increase and decrease of osmotic pressure of guard cells. When the guard cells are fully charged with water, that is they are turgid, the guard cells pushed out, it bend outwards, opening the stoma. And when the guard cells lose water, they shrink and they come closer to one another, then closing the opening stoma. The osmosis also play a key role in the growth of radicle and plumule during germination of seeds. That is due to osmosis, when the germinating seeds imbibe water, the seed becomes turgid, it swells up, turgor pressure is developed, and that pressure is applied on the walls of the seed coat. As a result, it bursts open. The soft organs of plants like leaves, flowers and fruits and young stems are able to keep themselves stretched and stiff due to turgidity of their cells, which in turn also depends on osmosis. Children, I will be doing till here today. The remaining part of the chapter I will discuss with you next time, and tomorrow with this video, I will be uploading I will be uploading this video with the assignment. Up till here, you go through my videos and the key points also I will give in the assignment. Read those key points, learn them, and accordingly you do the question answers. The remaining part of the chapter which is left, I will be taking step by step next time, and then after completing the chapter, that assignment on that part will be given to you afterwards. So there will be two assignment I will give you in two parts. The assignment of this chapter will be given in two parts. Tomorrow you will get the first part, and afterwards you will get the second assignment of this chapter. Thank you.